Hello. So we'll just wait for 1 or 2 minutes for others also to join and then we'll start the session, okay? In another 2 minutes, we'll start the class, okay? Okay, let us start today's session. So, a very good evening, everybody. Just let me know that is my voice clear? Am I audible? Can you hear my voice? Okay, thank you. So, hmm, today we'll be doing RNA splicing, export, and stability. It is week 5 only. I don't know why it is written week 6. Okay, fine. So, we'll be covering the courses which are covered in the previous week, which is RNA splicing, export, and stability. So, first, let us start to quickly summarize what we have learned in these lectures, and then we'll come to the questions. Okay. So, lecture 1 was about the relevance of introns. That what is the importance of introns? So, introns earlier were known to be called as junk DNA. Okay, earlier people used to call introns as junk DNA. Why they used to call it as junk DNA? Because introns does not had any functions. They thought that introns do not have any functions because intron is getting removed during the splicing process, and it is only the exons which are attached and they are important in coding of the protein. So they used to call it intron as junk DNA. But over the period of time, it was known that indeed introns are very important and introns are responsible for the regulation of the expression of genes, that how much a gene is going to express. The expressivity of the gene is decided by the introns. Okay, so what I can say is that expressivity of the genes is decided by the introns. So now it is known that indeed introns are Very much important, equally important, sometimes even more important, because they decide that how much a gene is going to be expressed. And not just the expressivity of the genes, there are many more functions of introns which we'll be seeing in the subsequent lectures. So now coming to like how the introns have evolved over during the course of time. So the average number of introns per gene is higher in larger organisms than in lower organisms, which means that what humans, okay, so humans, even in mammals, even in vertebrates, humans are the most evolved organism, right? So humans have the highest number of introns, and the average number of introns uh, is subsequently has subsequently increased as the organism's complexity has increased. Okay, the number of introns range from 1 to 636, and the average size of the intron is about 150 nucleotide, and this is the size of intron about 3,000 nucleotide to up to 800,000 nucleotides. So this is okay, that this is the average number of introns and this is the size and everything, and you can see here this is the rate of transcription. You see, E. coli में the rate of transcription is 10 to 100 nucleotides per second. Homo sapiens 6 to 17 nucleotide per second. So this is the transcription rate and this is the translation rate among the organisms. Okay, this is fine. So now the question arises. Earlier the question how it arose was that if you are saying that if people used to say that introns are junk DNA and they are already getting spliced during the maturation, then why do genes actually need introns? What is the need of introns? So see, introns, to maintain introns in the DNA. We all know that everything, each and every sequence, each and every nucleotide which is added requires certain amount of energy to be present there. Okay, whether it's just a small stretch of DNA or a few nucleotides, to be present there requires ATP. So many introns are there in our genome. Our genome is filled with introns and exons. That means it requires a high cost of maintaining the introns. High cost of maintaining, that means it requires a lot of ATP to maintain the introns. And also, we need another complex machinery, which is the spliceosome complex or any kind of splicing machinery to remove the introns during transcription modification, and introns do not have any role also. Then why introns are at all needed? Why introns are important? Because there is so much high cost which is required for maintaining the introns in the genome. Then you are removing it also while we are post transcriptionally modifying the mRNA. Then why at the first place introns are important? Why introns are needed? This is the question that should come in your mind. So why introns are important? Because it allows the expressivity of the gene. Like I told you, introns helps in the expressivity of the gene. It allows the expressivity of the gene. Introns dilute the genes, especially the one which are prone to mutation, thus reduces the chances of mutation in a gene. It reduces the chances of mutation in a gene. How you think that introns can reduce the chance of mutation? Suppose if this is particular a region which is very much prone to mutation, okay? This area is prone to mutation because it is currently getting like whatever for whatever reason. There are exons, exon 1, there is intron, and then there is exon 2, and then again the intron 2 starts. Now in this area, if there were no introns, there would have been exon 1, exon 2, exon 3, exon 4, and so on. So now because there is an intron here, so the number of exons in this area has got diluted. There are only 2 exons which could fit in this area, whereas 4 exons can fit in this area. So the chances of mutations are lesser in this case. Why? Because here only 2 exons can get mutated, but here 1, 2, 3, 4 exons can get mutated. So It is diluting the effect of mutation. So introns dilute the genes. They are diluting the genes because in this particular area here, if there were no introns, four exons could have fit. But here only two exons have fitted because there is intron here. So it has diluted the genes, especially which are prone to mutation. So it introduces the chances of mutation. Okay, I hope this point is clear. Some pre-mRNA can be spliced in more than one way, producing isoforms. This we have already studied in the last class about alternative splicing, that one mRNA can give rise to one premature RNA, or one primary mRNA can give to many different kinds of mature mRNA. How it can give rise to many different kinds of mature mRNA? Because of differential splicing or alternative splicing. So because mRNA can be spliced in more than one way, therefore isoforms, mRNA isoforms can be produced. And if mRNA isoforms can be produced, which means that introns are again important. Introns increases the transcriptional outputs and translational outputs as well. So these are the few points which tells that why introns are important and why our body is keeping this high maintaining introns in our body, even if it is getting spliced out during post transcriptional modifications. Is it clear to everybody? Okay. So why genome need to have introns? For alternative splicing, because alternative splicing can only give rise to different isoforms of mRNA. Alternative splicing, which can give rise to mRNA isoforms. Introns promote transcription and gene expression. Transcript fidelity via nonsense mediated decay. This we'll see. Exon shuffling and increases recombination to create more products. I hope this is clear to everybody. So this was in lecture 1. So what we'll do is that, because this week's lecture is not much to explain. Much to explain means that the content which is there, some of them are very much self-explanatory, which need not much of the explanation, and some of the contents which are very detailed that I cannot cover in these 2 hours. So I will just kind of try to give you a gist of it, so that we'll spend more time in questions also. Just a minute. What happened? Okay, fine. Then lecture 2 was about introns in RNA splicing. So we have already studied that this point we have already studied. Then why genome need to have introns? Intron mediated transcriptional regulation. So we know that introns helps in the transcription regulation. Okay, so this transcriptional regulation can be splicing dependent also, and this transcriptional regulation can be splicing independent also. So let us see what is splicing dependent transcriptional regulation and what is splicing independent transcription regulation. So splicing dependent means U1 snRNA, which binds to the 5' splice sites, interacts with the general transcription factor TF2H. And splicing independent means presence of enhancer promoter element in the intron. So what it is saying is that how introns helps in transcriptional regulation. That how much transcription will happen, kitna transcription hoga, how much would be the efficiency of transcription, how introns decide that. Okay. So some kinds of enhancer and promoter elements are present in introns. And if what happened? Okay, just a minute. Why is not working? There is some problem, that's why it's not working. Ah, now it started working. Okay, fine. So splicing independent, how we can say? Because this intronic sequence can have some enhancers or promoter elements. And these enhancer promoter elements will increase the rate of transcription, or you can have some other sequences also which can decrease the rate of transcription also. But the point is that these elements are present in the intronic regions. This is a splicing independent way. And what is splicing dependent way? Splicing dependent means that for the removal of introns there is a splicing process, and for splicing some snRNA comes into picture. So this U1 snRNA which comes during the splicing process, it interacts with the general transcription factor TF2H, which means that while splicing happens, the enzymes which are important, the enzymes which are required for splicing also helps in binding with this general transcription factor, which is TF2H. So this is how introns helps in the regulation of transcription. They helps in the regulation of transcription via splicing method, splicing dependent method, or via splice. Just a minute. Where is it? Hmm. Via splicing dependent method or splicing independent method. If they are doing a splicing dependent method, then what would they do? Then the enzymes which are required for splicing, which is U1 snRNA. So this U1 snRNA, along with doing the splicing of introns, it also binds to a general transcription factor, which is TF2H, and helps in the transcription. So how splicing independent method? Because this introns can have sequences such as enhancer sequences or promoter sequences which increases the rate of transcription. So this is how genomes need to have introns, and introns are indeed important for the expression and for other factors also, for the transcription regulation overall. So now why gene need to have introns? There are two general pathways that we need to study. Nonsense mediated decay pathway. Okay, now what is this nonsense mediated decay pathway? So this pathway actually um, degrades the mRNA which contains premature translation termination codons. So nonsense, from the name itself you can see that nonsense mediated decay. If you know, if you have studied mutations, you know that there are various different kinds of mutations that takes place. Okay, there is missense mutation, there is nonsense mutation. Okay, what is missense mutation? That instead of your nucleotide, any other nucleotide has come, and now the sense of the codon has completely changed. Nonsense mutation, what happens? The change in the nucleotide lead to the formation of any kind of stop codon. So there would be premature termination of mRNA. Because of nonsense mutation, there would be premature termination. Premature termination. This is your nonsense. So nonsense mediated decay pathway means that suppose where if there is any nonsense mutation that has happened, and because of that nonsense mutation there would be premature termination of mRNA, because that is what nonsense mutation means, that whenever there is a nonsense mutation, because the actual codon has now changed to a stop codon, therefore the translation would be terminated prematurely. Okay, so this decade pathway, what it does is that it degrades such mRNA which have a premature translation termination codon. Could you understand? That nonsense mediated decay pathway, what it does is that it degrades such mRNAs which has premature translational termination codons. And this nonsense mediated pathway is more efficient if your stop codon is present upstream, means before of the exon junction, and this is very important. Then next is exon shuffling and increased. So this nonsense mediated decay pathway is supported with the help of introns. Okay. And then exon shuffling and increased recombination. So we know that because we have different introns, and exon shuffling creates new combination of exons by intronic recombination. Exon shuffling was vital in creation of multi-domain genes in metazones. Introns increase recombination rate in coding regions of genes. So what does it mean? It means that because there are so many introns present, and therefore which introns are getting spliced out and which introns are getting not spliced out, or how the shuffling of the introns takes place, they kind of decides what would be the sequence of the exons. So exon shuffling is possible because of the different intronic recombinations that happens. Okay, so therefore exon shuffling, which helps in new recombination and new variations, is possible because of introns. So introns are helping in creating variations. Okay, it creates introns, creates variations. Okay, then comes the what is the splicing cycle. So we all know that splicing takes place in the premature RNA, in the pre-mRNA, and then it forms the mature RNA. So this image tells us how the splicing takes place. Okay, so this is the whole process, and how there is, this is one exon, this is one exon, and in between there is intron. Let us try to just quickly see this. This is exon 1, this is exon 2, and this is the intron, and this intron has to removed. So there are two enzymes, U1 and U2. So first U1 and U2 comes, and U1 binds to the 5 prime exon, U2 binds to the 3 prime exon. Always remember this. U1, it binds to the 5 prime exon, and U2, it binds to the 3 prime exon. Okay, and then when U1 and U2 comes and binds to 5 prime and 3 prime exons respectively, then other complexes also comes and joins in. So this complex is known as complex A. Okay, when only U1 and U2 are there, this complex is known as complex A. Now when U5 and U6, U4, which are together, when they comes and join this complex, then complex A is converted into complex B. Okay, you see this is U1 and this is U4, and this U6 U4 sorry U2, and there is U6 U4, which is a combined complex, and then U5 also comes and join. Now it has become a complex B, and then what happens, this U4 and U1 are removed. So what happens, this U6 comes and binds, and U2 comes and binds. So this is a B star complex. Now it has come even closer. You see, this is kind of stretching it to come it to closer, and when it stretches it to come it to closer, it kinds of forms a loop. The intron kinds of forms a loop, which is complex C. And when the introns complex intron forms a loop, then sequentially this intron is excised as a lariat structure. Okay, so intron is removed as a lariat structure. Okay, so this is how in general how the splicing takes place. Now there are three classes of RNA splicing, which depends on the kind of introns which are involved. Okay, let us quickly see this. I'm sorry, I'm also eating in between because um straight away from my class I'm coming. So I mean from my lab I'm coming. So I haven't had anything to eat and it's a 2 hour class. It's a long class. So I also need some energy for to teach you guys. So yeah, yeah. Anyways, so yeah, there are three classes of RNA splicing, which depends on the kind of introns which are present. So nuclear pre-mRNA, which we talk about the mRNA splicing, which is the most common, which is used for eukaryotic genes, and the mechanism is two sequential trans-esterification reaction takes place, and at the branch site there is A and spliceosomes are involved. This is the one that we generally study. Whenever we talk about whenever we talk about splicing, we talk about this nuclear pre-mRNA splicing, which is the most common. What we need to study are the group 1 and group 2 introns. What are group 2 introns and group 1 introns? They both are very rare. Group 1 introns are present in nuclear ribosomal RNA in some eukaryotes, some of the organelle genes, and some prokaryotic genes as well. Here in here the branch site is A, here the branch site is G. Okay, so group 1 introns is majorly present in rRNA, and here also two trans esterification reaction is required, but the branch site is G. Here the branch site was A, and group 2 introns it is found in some eukaryotic genes and genes from organelles and prokaryotes, and here the mechanism is same as pre-mRNA. That means two trans esterification reactions are required and branch site A, and RNA enzyme coded by intron. So here ribozymes are used. So in group 2 introns, ribozymes are used. This is important. You should remember that in which category of introns ribozymes are used. So in group 2 introns, ribozymes are used. Fine. So this is about generally like the basic mechanism that I wanted to discuss about the splicing. Just a minute. Okay. Now comes another concept which we need to study a bit, that is important in this are cis splicing and trans splicing. So what is the meaning of cis and trans? Can anybody tell me? What is the meaning of cis and trans? Hmm? Cis means in same, trans means in different. Like trans factors, something which is coming from outside. Hmm? Cis means within.
This means from in and trans means from outside.
Hmm. Good. So when we talk about trans splicing, that means something is missing in the cis splicing complex, cis splicing machinery, and something is coming from outside, which helps in the splicing of the exon. Okay, now you see here, if you see these two figures, you can tell that there are two exons, exon 1, exon 2, and then your U1 and U6 comes. U1 binds to the 5' splice site, U2 comes and it binds to the 3' splice site, and then in general, like the normal splicing that happens is your cis splicing. What if you have only one exon here? And then if you want to remove something, if you want to remove the non-coding part, the non-coding part in this case is not known as intron. The non-coding part in this case is known as outron. Okay, this is the non-coding part in this case is known as outron. What happens here in these kinds of cases, when there is only one exon, the exon in the front is missing, then leader sequence, externally a leader sequence which is SL1 SNRP comes, and it kind of provides the 5' splice site which is earlier provided by the provided by this exon. So this exon was used to provide the 5' splice site. But since there is no exon present here, so leader sequence which is coming from externally. This is an external factor which comes. Okay. So this is an external factor which comes, and this external factor is nothing but a leader sequence. It's a leader SNRNP. It is a leader sequence which is SL1. So this leader sequence provides the 5' splice site, and then splicing takes place. So cis splicing means the 5' splice site is provided by the exon. Trans-splicing, because there is no exon here, so the leader sequence from externally it is coming and it is providing the 5' splice site. Okay, so I hope this is clear. Okay, now we come to lecture 3. So this also talks about the same thing, the cis and the trans splicing that we have talked about. So you see, this is SL means splice leader RNA. Okay, SLRNA. So use of splice leader RNA in trans splicing is a mechanism of gene expression, and it is a form of RNA processing in which splice leader sequence. So because there is no exon which is present, therefore externally a splice leader sequence comes and it joined to a 5' leader-less pre-mRNA to form the mature 5' end of the RNA. Because earlier what happens, if you have exon, then this exon will only provide the 5' end where U1 will come and bind. But if this exon is not there, then this splice leader sequence, SL sequence, comes and it provides the 5' end. This process adds a conserved sequence to the 5' ends of mRNA and brings the 5' cap to the mRNA. So this is why trans splicing is important in such cases. Okay, this is what we have just studied. Okay. So here it says that cis splicing involves two exons. See, exon number 1, exon number 2. The two exons which are joined by an intron, and so cis splicing means two exons are joined by eliminating the intron, while trans splicing involves using a leader segment from one source and joining to the 5' end of another pre-mRNA which lacks the 5' leader sequence segment. So some some pre-mRNA which lacks the 5' leader segment, in those cases trans splicing happens, because this splice leader sequence, it provides the 5' end. Okay, 5' splice site. So therefore that is why trans splicing is important. And in trans splicing there is no 5' splice site on the pre-mRNA, because in the pre-mRNA no such site is present. Therefore 5' splice site is provided by the donor SNRP, which interacts with the U2, and this it is provided. The region between the 5' end and the trans splice site is called as outron. This is your outron. This is intron, this is outron. So I hope this is clear to everybody. Just a minute. Hmm. So trans splicing ka function. So what are the functions of trans splicing? It regulates gene expression, 5' cap provided by the splicing leaders, and helps in the separation of cistrons. Okay, and Uh, okay. Now, spliceosomes. Now, we have talked about the different kinds of splicing that takes place, cis splicing and trans splicing. What is the difference between cis splicing and trans splicing? In what conditions your trans splicing takes place? We have also studied about why introns are important, why genome needs introns, and how introns have evolved with the complexity of the organisms, and how this mRNA splicing cycle occur, what are the factors that are involved in this mRNA splicing cycle, that also we have covered, okay, right now. Then we started with cis and trans splicing. Now we'll come to the spliceosomes, because here we are talking everything about splicing. So spliceosome complexes can be of two types, okay. One is U2 type intron mediated, and second is U12 type intron mediated. U2 type intron mediated splicing when happens, then this spliceosome is a major spliceosome, okay. U2 type intron spliceosome is a major spliceosome, whereas U12 type intron spliceosome is a minor spliceosome, okay. These are the two types of spliceosomes, major spliceosome and minor spliceosome. What is a major spliceosome? Spliceosome, which comprises of 5 snRNA and more than 170 proteins. Spliceosome size is similar to the size of the ribosomes. RNA splicing is ATP driven. RNA participates in splicing reaction and all this. This is your major spliceosome, which is U2 type driven. I hope this is clear, which is more important to study. Then we come to another important point, which is SMN complex in the lecture 4. I think lecture 4 is the last lecture. Yeah. Yeah, then we'll be doing the questions. This is the last lecture which was covered in your previous week. I mean, we are not in the whatever it is, and then we'll do the questions. This is your RNA splicing. This is how RNA splicing takes place. Okay, so let us see the how many ATP and how many GTPs are gained. I'll give you 2 minutes. See, just try to see this cycle and analyze how many ATP and GTP are generated, and whatever I have written, whether it is actually correct or not. See it and tell me. Hmm. Count the number of ATPs and GTPs.
It's 10 ATP and 2 GTP. It's correct.
Hmm. Okay. So, see here. This is your exon. This is your exon 2. This is your exon 1. Okay. And this is your exon 2. Okay. This is the 5 prime splice site. This is the 3 prime splice site. This is the sequence that I have told you. It's a conserved sequence. We should remember it. At the 5 prime, there would be GU. At the 3 prime, there would be AG. GUAG or GTAG. And in the middle, this is a branch point. Okay, this is 5 prime splice site. SS means splice site. Here it is 3 prime splice site, and this is branch point, which is A. In nuclear pre-mRNA splicing, the branch point is A, whereas in group 2 introns, it was G, I think. Yeah, group 1 intron, it was G. Anyways, so now what happens? First, U1 comes and U1 binds at the 5 prime splice site. U1 will bind to the 5 prime splice site, and then U2 comes and U2 will bind to the 3 prime splice site. U2 comes and it binds to the 3 prime splice site. Okay. Just a minute. So now you see here two ATPs are needed. This is your pre-spliceosome complex. Then when these factors comes and bind, then again there is ATP, and then when these factors come bind, then there is ATP, GTP, and it all comes. So there is 1, 2, 3, 4, 5, 6, 7, 8, 9, and 10. So 10 ATP and 1, 2 GTP. So overall, in this process, 10 ATP and 2 GTP are used. So this is, you should know that in one RNA splicing, 10 ATP and 2 GTP, that means it is such an energetically expensive process. RNA splicing needs a lot of energy. But even then, when, if there would have been no use of introns, then why would our body would have evolved in such a way that it would every time for just one splicing reaction it is sacrificing 12 ATP. That means that introns are so much important to be present in the genome, in the DNA, that it can bear the cost of this much ATP for even one splicing reaction, because our body has evolved in such a way and nature always prefers where there is low expenditure of energy. Okay, and wherever there is no use, the that particular organ, that particular transcription factor, that particular any molecular component is vanished, so that ATP is secured. Okay. Okay, this is just that just a figure to tell you that how many these things are there. Okay. So these are like whatever I have written some things over here, that how this the mechanisms of how it is happening. I'm not going to spend much time over here because these are not that much important, but I have written the explanations here, that how U1 comes and binds to the intron exon boundary, because it is more or less the same thing. You see here what I have written. U1 binds in the intron exon boundary. Yes, we have already studied that. At the branch point U2 comes and bind. The branch point A is projected out, does not participate with U2 as an RNA, so that it can easily participate in trans-esterification reaction. And siRNA rearrangement takes place during splicing. So if you have gone through the lectures, just what I have written little explanation so that you can understand that how it is happening. Okay, just a minute. Sorry. Actually, my experiment is also going on, so my students are just updating me the progress that they have made. Okay. So now here also snRNA rearrangement in spliceosome. So how snRNA is rearranged? So you can see that U1 is replaced by the U6 helicase. So U6 helicase actually replaces the U1, and U6 brings U2 close also, and branch point bounded by U6 comes close to 5' exon. Basically, you should understand that if this is your U1 and this is your U2 and this is your intron, so this has to come close by. Okay, this has to has to be a contraction, so that they come close by, and first transesterification reaction also takes place, and also it can go as a lariat structure. So this all explains that how it should come to close by so that the transesterification reaction can takes place. Nothing much to explain, not to grow into detail. This is again, you can just read it out. I have just given the screenshots of the different complexes that happens, the detention complexes and the SM proteins. Not much to explain here, you should read it out. Okay, just you should know that SMN proteins is important. Normally it is important for the maintenance of specialized nerve cells which are known as motor neurons. Okay, this uh, is huh, this is what is already given, and this is something which is there in the lecture, not explaining much, that's why, because it is already present. Just for your brief clarification, that snRNA, small nuclear RNA, they are found to be tightly bound to several small proteins. And these small proteins are known as SM proteins. So the small nuclear RNAs are bound to some proteins, and these proteins are known as SM proteins. And these small nuclear RNA, which is snRNAs, are complexed with the SM proteins with other proteins to form snRNP. Okay, so just remember that snRNA, it bounds to some SM proteins, okay, plus some other proteins, and ultimately it forms snRNA RNPs, means RNA protein complexes, ribonucleoproteins, okay. And by mid 1980s, it became clear that these snRNPs help form a large complex which is known as a spliceosome around pre-mRNA, which is helping in splicing. So these snRNPs actually are responsible for the formation of spliceosome complex. Okay, and what is this spliceosome complex? This spliceosome complex is actually the complex which has all the U1 and U2, all the enzymes, which is actually responsible for the spliceosome or for the splicing. So this spliceosome complex is formed of snRNP, and these snRNPs are formed by snRNA plus SM proteins and other proteins. Okay, I hope this is clear to you. That snRNA along with some SM proteins, which are small proteins which are known as SM core proteins or SM proteins. So snRNA along with some SM proteins and some other different proteins, it forms a complex which is known as snRNP, and these snRNPs ultimately combines to form the spliceosome complex. Okay, then several modifications. After several modifications, the spliced pre-mRNA get transformed to mRNA, which is exported from the nucleus and translated into proteins by ribosomes. Okay, that is all fine. So these SM proteins, if we talk about what are these SM proteins, so these SM proteins are actually a group of proteins which are present in neurons. It is present in spinal cord, and it is responsible for the maintenance of motor neurons, and these cells are located in the spinal cord and the part of the brain that is connected to the spinal cord. Okay, and so basically these SM proteins plays an important role in processing the mRNA, and which serves as a genetic blueprint for making protein. So this means that these SM proteins are not only important in your mRNA processing, but they are also present in your spinal cord, and they help in the maintenance of motor neurons. Okay, so there also SMN proteins are important. Then comes how this as a core complex, assembly of core complex happens. So this is just a mechanism, you can just go through it. I don't have that much time because I have to cover the questions also. Then we come to What are the mechanisms of introns gain and intron losses? Okay, so how intron gain and intron loss happen? You see, we already have studied. This is transcription happens, splicing happens. This is the intron which is after splicing is removed from the as in the form of a lariat structure. Then reverse transcription happens, and this is reverse transcription, and this is your cDNA. This is your cDNA. And then this is your genomic DNA and this is your cDNA. So when there's a recombination with the genomic DNA, what happens? This particular region of the genomic DNA which has intron, that also gets deleted, or some part of the intron gets deleted during the recombination process, and therefore some of the intron is lost. You understood? When this cDNA, complementary DNA, when it recombines with genomic DNA, at that time what happens, some part of the intron is lost, which is shown here as genomic deletion. Therefore there is some loss of the intron which takes place. This is how intron loss or intron gain happens. Okay. I mean, this essentially talks about the intron loss, that how intron loss happens. So this is the cDNA. So you should know that when cDNA, it has recombination with the genomic DNA, then what happens? That some part of the intron is lost, and therefore there is intron loss that happens. Okay, so now we have completed the questions. Now I mean the theory. Now we'll come to the questions. Okay, we'll come to the question part. For each question, I give you 2 to 3 minutes. If you know the answer, you write it in the message box. After 2 or 3 minutes, depending on the question, I'll switch the screen and then we will discuss the questions and the answers. I hope this is clear to everybody. So the first question is, SMN complex is involved in RNA splicing, maturation of snRNP, formation of snRNA isoforms, or snRNA duplication. This is your first question. Okay. So it's right now it is 7:52. I will switch the screen after 2 minutes. You have 2 minutes and try to understand the question and answer it in the, just type your answer in the message box and then I'll see. Okay. Do it. One more minute left. One more minute left. Okay. I'm switching the screen now. Hmm. So Guna has answered RNA splicing and Sundar Raj has answered maturation of snRNP. Okay. So the correct answer is maturation of snRNP. So why that is the correct answer? What is the question? The question says SMN complex is involved in RNA splicing, maturation of snRNP, formation of snRNA isoforms or snRNA duplication. Okay. Now, SN complex, it always plays a crucial role in the first of all assembly of the spliceosome snRNP and how it helps in the assembly of spliceosome snRNP by helping in the transport of snRNP and biogenesis of snRNP to the nucleus. And this process is very much important for the proper RNA splicing, but this process is not directly responsible for splicing or for a formation of RNA isoforms or for RNA duplication. Okay, so what ultimately SMN complex do, ultimately it is helping in RNA splicing, no denial in the fact. But how it helps in RNA splicing? Because first of all it helps in the biogenesis and maturation of snRNP, which helps in the formation of spliceosome complex, and then it helps in splicing. Okay, so directly, if there would not have been this option maturation of snRNP, then you can have chosen RNA splicing, but this is more direct. First, SMN complex helps in the biogenesis and maturation of snRNP. These snRNP then helps in the formation of spliceosome complex, and that spliceosome complex helps in RNA splicing. So, like that, we have to look for the answer which is most relevant. Okay, I always say whenever we solve multiple choice question with students, I always say that MCQs can be confusing. Same option can be right in one question and same option can be a wrong statement in another question. It is all comparative. In this case, this RNA splicing is a wrong statement. But suppose if there were options RNA splicing, snRNA duplication, uh, transcription initiation, none of the above. Now in this case, RNA splicing is the correct answer. So in MCQ, you not need only to look for the correct answer. You should not only see why this is correct. You should also see why others are wrong. This is very important when you're solving MCQs. Not only why your selected option is correct, but why others are also wrong. Because same point can be right in one question, it can be wrong in other question. It is all comparative. Okay. Okay. Next question is the RNA component deleted from the pre-mRNA after trans-splicing is called as A. Lariat structure, B. Polypyrimidine, C. Outron, or D. SL sequence. Okay, so we have to answer that. We have already studied what is a cis splicing and what is trans splicing. So there you have to tell me. So I'm giving you 2 minutes. Then I will switch the screen. Okay, do it. One more minute left. Okay. I'm pushing the screen now. Hmm. So both have answered outron, and it is the correct answer. Outron is the correct answer. So the RNA component which is deleted from the trans splicing is outron. We have already studied that mm, when it is a trans splicing, it is known as outron. Okay. Intron is only in the case when it is cis splicing, and intron is also removed as a lariat structure. Okay. So, just a minute. I will put like this, otherwise. So why not lariat? Because it is formed during the cis splicing. Lariat structure is formed during the cis splicing. This polypyrimidine, so polypyrimidine tract is present in introns, which helps in splicing, but it is not specific to trans splicing. Okay, this polypyrimidine tract helps in splicing, but it is not specific to trans splicing. It is not specific to trans splicing. Outron is the correct answer, and SL is the sequence which is added during trans splicing. It is not removed. Okay, this is a splice leader sequence. Splice leader sequence which is added during trans splicing and not removed. Okay, so I hope this question is clear to everybody. Next comes, which of the following are involved in RNA splicing? RNPs, RES complex, exosome, or DCP2? I am repeating the question. Which of the following are involved in RNA splicing? RNPs, RES complex, exosome, or DCP2? Answer it. I will switch the screen after 3 minutes. Okay. Try to answer it by then. 2 more minutes left. One more minute left. Okay, 30 seconds. Okay. So both Guna as Sundaraj has answered RES complex, and it is the right answer. Okay, good. So what is this RES complex? This is hmm, this RES complex. It is directly involved in RNA splicing by enhancing the efficiency of intron removal and intron definition. So basically, this how intron removal happens, this efficiency is very much improved with the help of this RES complex, and therefore it is the correct answer. Good. Then the next question is, which of the following results from mutation in splicing factor? Okay. First is muscular dystrophy. Second is cystic fibrosis. Third is spinal muscular atrophy. And the last option is dementia or Parkinson. Okay, repeating the question. Which of the following results from mutations in splicing factors? Muscular dystrophy, cystic fibrosis, spinal muscular atrophy, or dementia or Parkinson? Try to answer this question. You have 3 minutes with you. In 3 minutes, I will switch the screen, okay? It's 8:08 right now. I will switch at 8:11. Answer it. Try to understand each and every option. Try to negate the wrong ones. 2 minutes more. Cystic fibrosis, you should know the reasons why cystic fibrosis happen, why dementia or Parkinson happens, why muscular, what is the reason behind muscular dystrophy, what is the reason behind muscular atrophy. If you know the reasons behind all these things, it is easy for you to negate the wrong options also. Okay. 1 minute more. Okay. And now I'm switching the screen. Let us see what you have answered. Okay, so Guna has also answered spinal muscular atrophy, and Sundar Raj has also answered spinal muscular atrophy, which is the correct answer. Spinal muscular atrophy is the correct answer. Okay, so this SMA, which is spinal muscular atrophy, it results from a mutation in the SM proteins. SM proteins we have, uh, SMN proteins we have discussed. So there is a mutation in SMN1 gene, SMN1 gene, which is survival motor neuron 1. Survival motor neuron 1. SMN stands for survival motor neuron 1, which encodes for the SMN proteins. So this SMN1 gene gets mutated, and this SMN1 gene codes for the SMN proteins, and we know that SMN proteins are essential for the assembly of snRNPs and ultimately the formation of the spliceosome complex, which helps in the splicing. So defects in SM1 lead to widespread splicing errors, and therefore, this is one of the reasons. Now, why not other options? So, this SMA is spinal muscular atrophy. It results from the mutation of SMN1 gene, and this SMN1 gene codes for SMN protein, which helps in the coding of spliceosome complex and splicing. Now this muscular dystrophy, it is caused by mutation in DMD gene. Some DMD gene gets mutated and it affects the dystrophin protein, which affects your dystrophin protein, but not the splicing factors. Cystic fibrosis, we all know, I mean, you should know that cystic fibrosis results from the mutation of which gene? Which gene is affected in cystic fibrosis? CFTR gene, which basically affects the ion transport and not splicing. Okay. Then dementia and Parkinson disease. These are neurodegenerative diseases. Okay. These are neurodegenerative diseases. Neurodegenerative diseases. So these are neurodegenerative diseases which is and they're not directly caused by the mutation in the core splicing factor, although these are involved in splicing defects. Here also there are some splicing defects which are involved, but these are not directly caused by mutation in the core splicing factors like SMN1. So therefore the correct answer is spinal muscular atrophy. Next question is, transposomes play a role in exon deletion, intron deletion, exon gain or intron gain. Okay. Transposomes. Think. Right now it is 8:15. I will switch the screen after 2 minutes. At 8:17 I'll switch the screen. Okay. Meanwhile, think of the correct answer. First of all, you should know what are transposomes and then how transposomes can help in any of this function. One more minute left. Hmm. Okay, time's up. Hmm. Intron gain. Okay. Yes. The correct answer is intron gain. So how intron gain is the correct answer? C. Transposomes are nothing but jumping genes. We call it as jumping genes, which can move from one place to another place. And they can move from one place to another place through various mechanisms, which we call as cut and paste mechanism or copy or paste mechanism. Cut and paste mechanism means that they cut from one place and then they get pasted to another area. Or copy paste mechanism, they copy and then they get pasted to some other way. Anyways, in any of the way, suppose from somewhere it takes one part and then it insert it to some other part. So transposomes can contribute to intron gain by inserting into genes and later it acquiring the splice signals which lead to the formation of new introns. Okay, and especially when we talk about this mechanism, we're talking about retrotransposomes, which can introduce sequences that can later recognized as introns by splicing machinery. This process plays a role in genome evolution also, and it helps in increasing the genomic complexity also. So the correct answer is intron gain. Next question is, RNA helicase has a feature called Hawk's box, dead box, E box or mad box. Okay, 2 minutes to answer this question and then I will switch to the screen. RNA helicase has a feature which is called as what? Right now it is 8:19. I will switch the screen 8:21. One more minute left. Okay, now I'm switching the screen. Okay, fine. Uh-huh. Okay, so huh. So Guna has answered dead box, and dead box is the correct answer. So RNA helicase has a feature which is called as dead box. What is the meaning of dead box? So dead box stands for a conserved amino acid sequence, which is aspartate, glutamine, alanine, and aspartate. Okay, this is the aspartate, glutamate, alanine, aspartate. This is a sequence which stands for dead. And RNA helicase, we know what are RNA helicase? These are enzymes that helps in the unwinding of the secondary structures. In the case of RNA, secondary structures, and they are essential for splicing processes. And these RNA helicase belong to the dead box family, which is named after the conserved sequence, which helps in the ATP binding domain. Now, why not other options? Because Hox box. Hox box is found in Hox genes. Okay. Hox box is found in Hox genes and which helps in regulating the body patterns. If you have studied in developmental biology, they help in regulating the body patterns during development. Okay, so this is a different option. Dead box is the correct option. E box. E box is a DNA sequence which is involved in transcription regulation, but not it is a function of RNA helicase. E box is also involved in transcription regulation only. Okay, E box is also involved in transcription regulation, but it is not a feature of RNA helicase. Then mad box regulate gene expression in plants and animals. These are transcription factors. These mad box are found in MADS box transcription factor. They are found in MADS box transcription factors and it regulates gene expression in plants and animals. So the correct answer is dead box. Next question is trans splicing do not rescue any type of RNA useless. True or false? 2 minutes to answer this question and then I will switch the screen. Try to answer it. Read the question carefully and then answer. One more minute left. One more minute is left, then I will switch the screen. Okay, now I'm switching the screen. Okay, so Guna has also answered true and Muthu Kumari has also answered true. Okay, and the correct answer is true. So, why it is true? So, question is trans-splicing do not rescue any type of RNA useless. So, trans-splicing is an important biological mechanism. It does not have a general function of rescuing all the non-functional RNA. Some RNA that are degraded due to mutations or some RNAs which have incorrect processing or some RNA which have some kind of structural defects that can be but rescued by or salvaged by trans splicing. Okay, it's not a universal. Trans splicing is not a universal RNA repair mechanism, but it helps just in salvaging some of the type of RNAs which are degraded due to mutations or some different processing and all. Okay. Then the next question is, minor spliceosome functions similar to major spliceosome on pre-mRNA. True or false? Minor spliceosomes, we have studied about minor and major, and you must have studied in your lectures also that minor spliceosome functions similar to major spliceosome or on pre-mRNA. True or false? Tell. 2 minutes. I will switch the screen after 2 minutes. One more minute left. One more minute left. Okay, now I'm switching the screen. Hmm. So, Guna has answered false and Muthu Kumari has also answered false. Minor spliceosome function similar to the major spliceosome. The correct answer is true. See, minor spliceosome, it functions similar to major spliceosome in a way that it removes introns from pre-mRNA through the splicing process, but there are some key differences. But whatever is given, given in the portal, they have said it's true. But I'll give you the similarities and differences both, and you can decide by yourself. You are better judge. So the similarities are that both perform the pre-mRNA splicing by excising the introns and joining the exons together. First thing. Then both use the snRNPs, that is the small nuclear ribonucleoproteins, to recognize the splice sites. That is another thing. And both follow a similar two-step trans-esterification mechanism for splicing. So these are the points that how uh, major minor spliceosome, it functions similarly to major spliceosome because both are performing pre-mRNA splicing, both are using snRNP, and both follow the two-step trans-esterification reaction. These are the three major similarities. Now, differences can be that major spliceosome, it removes U2 type introns, we have studied, and minor spliceosome, it removes U12 type of introns. And U12 type of introns are rarer. So the spliceosomes which are used by major and minor spliceosomes, they are also different. Major spliceosome use U1, U2, U4, U6. Okay. So the major one, U1, U2, U4, U5, U6. Minor, it uses U11, U12, U4 ATAC, U5, and all these things. So I think the answer should be false, because if they are spliceosomes, either they are major spliceosome or minor spliceosome. Spliceosome, that means anyways they are going to do the splicing now. So anyways they are going to remove the introns because they are spliceosome. So uh, since they are spliceosomes on pre-mRNA, and mRNA follows the trans-esterification reaction, so obviously they will have trans-esterification reaction. But the difference that makes them major and minor is the use of the this enzymes. That major uses the minor major, I mean the enzymes and the type of introns that it removes. So I think the answer should be false only. It's not correct. True is not the correct answer. But it is given in your, I mean, portal, it is given true only, but I think it should be false. Okay, next question is snRNA assemble site specifically on pre-mRNA. Is it true or false? snRNA assembles site specifically on pre-mRNA. True or false? Right now it is 8:32. I will switch the screen after 2 minutes. Okay. One more minute left. Okay, now I'm switching the screen. Hmm. So both Guna and Muthu Kumari has answered true. That snRNA assembles sites specifically on pre-mRNA, and it is the correct answer. So these snRNAs are, what are snRNA? These are small nuclear RNA. These are essential component of the spliceosome, and they assemble sites places specifically. So how they assemble sites specifically? We have already studied that first U1 comes and U1 bind to the 5' splice site. Then U2 comes, U2 bind to the branch point, and then U4, U6, and U5 snRNP comes, and then they form the spliceosome complex, and therefore it all happens. Therefore, there's a specific site at which all these snRNA components come and bind. Randomly, it doesn't come and bind and sit anywhere. Okay, it needs a specific site where it can come and it can bind to it. So yes, the correct answer is true. Okay. Last question is trimethyl guanosine cap, that is TMG, is not essential for nuclear import of snRNAs. True or false? Trimethyl guanosine cap is not essential for nuclear import of RNAs. True or false? I will give you 2 minutes. Right now it is 8:36. I will switch the screen at 8:38. Okay. You have 2 minutes to answer this question. 1 more minute left. Okay, now I'm switching the screen. Let us see what you have answered. Last question. Okay, so both Guna and Muthukumari has answered it false, and the correct answer is false. Okay, the question says trimethyl guanosine cap is not essential for nuclear import of snRNA. Okay, so it is false because it is indeed essential for the nuclear import. So how the snRNAs are transcribed in the nucleus and they are exported to the cytoplasm for maturation. During this maturation, the 7-methyl guanosine cap is converted into TMG, that is trimethyl guanosine cap. And this trimethyl guanosine cap, it serves as a nuclear import signal which allows the snRNA to be transported back to the nucleus. And without this TMG cap, snRNAs fail to efficiently re-enter the nucleus and cannot properly function in splicing. So what does it mean? That you have snRNAs. These snRNAs are transcribed in the nucleus and then they are exported to the cytoplasm maturation. During the maturation, what happens? Then 7-methyl guanosine cap is converted into trimethyl guanosine cap, which is known as TMG. And this TMG cap, it helps as a nuclear import signal, which allows the snRNA to be transported back to the nucleus. Okay, to be transported back to the nucleus. So what happens is that without this TMG cap, snRNAs will fail to efficiently re-enter the nucleus and then therefore they cannot function properly in splicing. So if we want these snRNAs back into the nucleus, then we need this TMG cap. Okay. So let us very quickly, in a minute or so, we'll quickly summarize of what we have studied. Okay, so first of all we have started that how the introns have evolved during due course of time and why, although that there is such a high cost of maintaining the introns and we need the splicing machinery, even then why introns are important. Because introns are important because they helps in the expressivity, it reduces the chances of mutation, it helps in producing the isoforms, and it increases the transcriptional and translational outputs also. So then how introns can help in transcriptional regulation? We studied that there are two mechanisms by which introns can help in transcriptional regulation. First is splicing independent, where introns can have enhancer and promoter sequences, or splicing dependent, where one of the enzyme which is responsible for the splicing of the introns, which is U1SN can also bind to the TF2H transcription factor and helps in the transcription. Then we have studied that the two different pathways which tells us that why genome need to have introns, nonsense mediated decay pathway and exon shuffling pathway. Then how the splicing takes place, how the various complexes are formed, and what are the different classes of splicing that takes place. We have studied that there is free mRNA class, which we already know, the most common class which we often study. Then there are group 2 and group 1 introns. Group 2 introns basically use the ribozymes, and group 1 introns also use the ribozymes. And only difference is that group 1 introns, they have the branch site as G. Two transesterification reactions are required, but they have the branch site G. Here you have the branch site as A. Then what is cis splicing and trans splicing? How, when there is no upstream exon present, then this leader sequence comes and it provides the 5' splice site, which helps in the proper removal. And this is known as outron. The part which is removed is not known as intron because it is not present inside, because there is nothing to be called as inside. It is outron. Okay, then this is what about the splice leader sequence, how it helps and it comes and it binds. Trans splicing, then the differences between cis splicing and trans splicing. Then what are the two types of spliceosomes, major spliceosomes and minor spliceosomes? What is the basic difference between the two spliceosomes? Is the type of intron. In major spliceosome, the type of intron is U2 type. In minor spliceosome, the type of intron is U12 type. Then how many ATPs and GTP are used during the RNA splicing, and this shows that in one cycle of RNA splicing 10 ATP and 2 GTP are used. Okay, so approximately 12 ATPs are being used in one RNA splicing, which is such an energetically expensive process. But still, if RNA splicing happens, means still RNAs are present, is itself states for the importance of RNA in our genome. That RNAs are so important in our genome that in spite that of the fact that it requires approximately 12 ATPs to remove intron during RNA splicing, still DNA contains the RNA introns. Then what are the different snRNPs which are present in the spliceosome complexes? And then we come to the SMN proteins and how the SMN proteins are formed. Then what is the mechanism of intron loss? And then we came to the questions. Okay, so this completes this week's lecture. So I hope you could understand. If you have any questions, you can ask me. So could you understand anything? Were you able to understand what I was trying to discuss here? Were you able to level up with the pace? Although I was trying to be slow only, I don't think I rushed that much. But were you able to level up with this with my pace? Okay, okay, fine. Good to know that. So we'll meet again on Tuesday. The timings are the same. 7 to 9 we'll meet, and then we'll discuss the next week lecture and the questions. So this is the way we are going to thank you Muthu Kumari. So this is the way which by which we are going to use do it. We'll just briefly discuss the theory of that particular week's lectures, and then we'll do the questions simultaneously. Theory lecture, theory questions we'll do, so that your concepts are also revised a bit, and also you are doing the question, which is actually the major part and the main part. Okay, then bye bye everybody. We'll meet in the next week class. Hmm. Okay. These are the slides. This is just the important point that I am highlighting for the people who are going to see the lecture. Okay, this is important. Where they have talked about that how 7-methyl guanosine cap is converted to this TMG, 2,2-trimethyl guanosine cap. Okay, this is important. That's why I'm highlighting it. Okay, fine. Hmm. Any other thing? Okay.